Hello, folks, and welcome to Cellular Respiration for AP Biology. This one is going to be a long one, so buckle up. Feel free to take it in segments. Listen to one part, take a break, go do something else, and then come back to it. But as I kind of have warned you in class, cellular respiration and photosynthesis in one of the next videos I record will be kind of a big explanation. There's a lot to it. The good news is, even though this seems really complicated because there's a lot going on, you still are not as complicated as it could be. Just keep reminding yourself of that. This is not a college level biochemistry class. You don't have to remember all the intermediate steps and so on. Instead, I'm going to keep referring your attention to the notes sheet that I put out with this for you in class or in Google Classroom. Or on my Weebly, where you are looking at kind of that chart with the overview of the processes, what's going in, where are we, what's coming out, and what's the point of this step. So we're going to really focus in on that as you do this. So we're going to start with the catabolic processes. Remember that catabolism, right here, catabolic processes, catabolism are the processes that break down molecules. And when we're talking about that, we are talking about breaking down complex molecules. And usually you're using this to sometimes put together simple molecules for the purpose of having energy later on. All right. Now remember, molecules have potential energy because of the arrangement of their atoms, and the ATP POGL that you all did together or did in class should have been a clue to that. You guys talked about that in the POGL. Also, what you have to remember in both photosynthesis and respiration, electrons move through these processes. And you're used to thinking in terms of chemistry where you have these redox reactions, right? Where the electron acceptor, the one that actually pulls an electron away from another molecule, is reduced while the electron donor is oxidized. But a lot of times where cellular respiration is concerned, if you take a look down here at this picture, what happens is it's not necessarily that you completely remove an electron. It's that an electron may move closer to one atom in a molecule than it was before. So if you look here in carbon dioxide, right, the electrons are closer to oxygen, right? But then if you look at the outcome here, you follow this arrow over to the other side to glucose, notice this pair of electrons has actually moved closer to carbon, which means carbon ends up getting reduced in that case. And again, here the oxygen is keeping the electrons close in water, but remember that water molecule is going to get released as oxygen gas. And this is in this particular reaction, which means in oxygen gas, the electrons actually move further away from each oxygen atom, which means that oxygen ends up being oxidized in this process. So just keep in mind that these are redox reactions. All right, so let's look at some of the players here in these reactions. ATP, right? There's our old friend ATP right there. You guys went through an entire POGL on this. I have had a chance to look at those POGLs, the ones that have been turned in so far. You guys have absolutely no misconceptions about this, right? I mentioned in an earlier video that the reason for ATP's existence is that it's a smaller molecule that contains a smaller amount of potential energy. And remember, the energy is in this bond right here, right? That holds together the last phosphate onto the molecule. That's about 7.6 kcals of energy, right? That you can find in an ATP molecule, right? And the reason for that is that glucose would have about 686 kilocalories of energy. So the whole point of cellular respiration is to break that glucose down. And as we release the energy from the chemical bonds in glucose, we create these ATP molecules, right? So that we're capturing that energy in smaller increments, because most of the processes that we do in cells don't require 686 kilocalories of energy. They require 10, 12, 8 kilocalories. And so you're not wasting as much energy by transferring glucose's energy to ATP. So in a lot of ways, cellular respiration is really about energy transfer. And one of the slides of notes that I didn't include here was reminding you about the law of conservation of energy, right? That energy is transferred, it can change form, but it's never lost. This is not a 100% efficient transfer of energy. I will tell you that. Not all of the energy from the glucose molecule is transformed into these ATP molecules. Some of the energy will be captured in the products of cellular respiration, right, which are considered to be waste, right? So they're not captured for the purpose of carrying out metabolism down the line. And then some of that energy actually is converted to body heat. So it's not lost. It's just no longer chemical energy that can be used to be harnessed to do work in cells. All right. Another important molecule that's going to come up here is NAD+. And here's a picture of what happens. Here's NAD+, and here is what happens when we allow it to be reduced to pick up electrons. It becomes NADH. Hydrogen atoms and electrons are going to be important for processes down the line. So as we break down the sugar molecule, one of the things that's going to happen is we're going to take this NAD+ molecule here, right? And we are going to convert it to NADH, and then we're going to use that. We're creating this specifically to destroy it later on in cellular respiration. But in the destruction of this molecule, right, or not really destruction, but what we're trying to do is take the electrons and the hydrogens off of it in the end, and that's going to be used to generate ATP. Now, in generating ATP, what we're doing is we're taking the ADP molecule that you see here with a phosphate, and this arrow, by the way, is reversible, and we're converting that to ATP. But again, you can actually phosphorylate when you go in this direction. When you take this equation in the opposite direction, you are hydrolyzing the ATP molecules. And again, a lot of that came up when you guys were doing your POGL, so I think we're good there. So just remember, I've got some notes here from the slides from class, but just remember that NAD molecule is really just there to carry electrons for us, because the last step is going to be an electron transport chain. Let's look at that. All right, so here's our cellular respiration overview, and again, as I was saying before, here's the electron transport chain, so hence the need for electrons. That last step in cellular respiration is going to need a bunch of electrons to work, and NADH is going to be carrying that. Plus, there's going to be another coenzyme that's going to be involved here later on, FADH2, and it starts out as FAD+ as well. Okay, so this slide is telling you basically an overview. Most textbooks will list that there are 3 steps. Really, there are 3 steps that generate energy. Okay, there's a 4th step. I call it a half step on your little sheet that you guys are filling out with what goes in, what goes out, and the point. Because notice it doesn't generate energy. See, step 1, energy, ATP. Step 2, what I've listed as the 2nd step, Krebs Cycle, energy. 3rd step, electron transport chain, energy. Notice there's no energy in this pyruvate processing step, so I kind of call it a half step. What this is going to do is get us ready to go from here to here, okay? But notice it is going to generate something important for us. It is going to generate some NADH, and again, that NADH is going to be useful over here. All right, so we've got 3 steps: glycolysis, and this is going to take place in the cytoplasm. So if you're looking at your chart, the where here is the cytoplasm, and then this pyruvate processing is going to take place as we move into the mitochondria. Okay, because the Krebs cycle is going to take place in the mitochondria. And then the electron transport chain is also taking place in the mitochondria. Krebs cycle takes place in the mitochondrial matrix. Electron transport takes place on the inner mitochondrial membrane. So we've got 3 basic steps, plus between step 1 and 2 is like a half step where we're getting ready for the next big thing, but it's not going to generate energy for us. All right, so let's look at glycolysis. And again, like I said, feel free to take each of these steps one at a time, to pause the video, to fill out your chart while you're going, just so that you're not lost in the process. All right, so glycolysis literally means sugar splitting. What you're going to do is you're going to take this glucose molecule and you're going to split it into 2 pyruvates. Okay, and you can think of a pyruvate as half of a glucose. Now, this takes place in 2 sets of reactions. So this is set 1 here, and this is set 2 underneath. In the first set of reactions, we call this the energy investment phase, because notice we're putting 2 ATP molecules into this reaction, right? So we're actually investing some energy to start to split the glucose. But then the second phase of this set of reactions we call the energy payoff phase. Okay, so here notice ADP is coming in and it's going to come out as ATP. Same thing here. Excuse me. So we are going to actually get a total of 4 ATP from this process. But remember, back in the investment phase, we had to spend 2 ATP, which means we have a net gain of 2 ATP. Okay. Now, notice also we've generated our old friend NADH, or maybe our new friend NADH, since you just learned about it today. NADH, remember, is going to be useful for the very last part of cellular respiration. So the whole point of going through these complicated steps, and notice I'm not talking about what's happening at each step. What I'm trying to get you to understand is in step 1 of these reactions, you invest ATP. In step 2 of these reactions down here, you gain ATP. In fact, you gain a net gain of 2 ATP, plus you're going to gain some electron carriers. One other thing I do want to talk to you about is in cellular respiration, there's 2 ways that you can generate glucose. Glucose, ATP, in glycolysis and in the Krebs cycle, you're going to see what we call substrate-level phosphorylation. In other words, you take an ADP molecule and some phosphorylated substrate in this poor little enzyme right here that's only got 1 active site and can only work on 1 molecule at a time, and you're going to transfer a phosphate group from here over to the ADP to create ATP. Okay, so this is substrate level phosphorylation. It happens in glycolysis. It's going to happen in the Krebs cycle a little later on. But it's really, really inefficient because these little enzymes can only work on 1 ATP molecule at a time. All right, so let's just take a sec and recap. Remember, glycolysis is in the cytoplasm. Whoops. Right? What's going in is glucose, right? So C6H12O6, and I'm really sorry, my handwriting is awful, I know. Now, in the process of doing this, we also put in 2 ADP, right? Or sorry, 4 ADP. Right, and in the payoff phase of this, we ended up with 4 ATP, right? But we also initially had to invest 2 ATP to get 2 ADP out. Now remember, this step is the investment, this step is the payoff phase, right? And the whole point of doing this was so that we got some ATP. and again, whoops, ATP, not APP, ATP, we ended up with a net gain of 2 ATP here, okay? Also, we put in NAD+, and that came out as NADH. Okay, so we ended up with a hydrogen carrier, and I believe that was 2. I'll have to go back and look, but I'm pretty sure that was 2 NADHs. So we've got some electron carriers, right? And remember that in the end, the glucose comes out as pyruvate. So we've begun to break glucose down here. We've generated 2 ATP. We've generated some electron carriers. All in all, great. Not very impressive in terms of energy, though, right? Okay, so we've started to break the glucose down, but we really haven't got a whole lot in terms of energy. Now, what is vitally important for you to understand, and you should probably draw a little red line here, is if there's no oxygen present in the cell, the process stops. This is anaerobic. The rest of cellular respiration requires oxygen to be present. Interestingly enough, we're not using oxygen here, we're not using oxygen here, but we are in the very last step. And so, in order to go on, you need oxygen. Okay? So even though, like I said, no oxygen is used in this step, and no oxygen is used here, we still need oxygen to be present in order to carry on, okay? So when we get to this red line moment right here, if oxygen is not present, then what's going to happen is you're going to go into fermentation. We'll get to that at the end of the video. And there's an actual point to fermentation besides making alcohol or lactic acid. But if oxygen is present, we will go on. And that's glycolysis. All right, so now we're at this little half step between glycolysis and the Krebs cycle. And to go further, the first thing we need to do is talk about the structure of the mitochondria. Notice you've got 2 membranes. Here's one membrane here in the mitochondria. That's the inner membrane that's all foldy-uppied. And then you've got this outer membrane. Okay, so the blue membrane here is the inner membrane, and then this one's the outer. Okay, the red membrane is the outer. And over on this side, this is just a colored electron microscope picture of what the membranes look like. All right, so the foldy inner membrane is the blue, okay? The space between the inner and outer membranes we call the inner membrane space. So that's the intermembrane space between the two. The folds of the inner membrane we call the cristae, and this space inside of the inner membrane we call the matrix. Okay. Now remember, glycolysis took place in the cytoplasm. The Krebs cycle is going to take place here in the mitochondrial matrix. Okay. But before we can get there, we have to take our pyruvate molecule that we have, right? And we have to do something to it. So as we move this pyruvate into the inner mitochondrial matrix, we're going to strip it of a carbon dioxide, and we're going to attach it to a little buddy, a coenzyme. Okay, so this coenzyme is going to help us to attach this pyruvate into the molecules of the Krebs cycle. That's why I call it a little buddy. It's just a little hand holder to kind of take the molecule where we need it to go and attach it in. So what we do is we convert this pyruvate into something called acetyl CAA, or if you're British, acetyl CAA. But notice we've taken this molecule, we've added an enzyme to it, we've taken pyruvate, stripped it of a carbon dioxide, so we're left with this acetyl group, and we attach it to a coenzyme A. And as I said before, no energy is generated in doing this, but we get a little NADH, which again, every little NADH counts because we need electrons later on, right? And NADH is carrying those electrons. So that's what happens in this half step as we move from the glycolysis in the cytoplasm into the matrix of the mitochondria. All right, so here's our little half step. We're going back here. So this takes place in the mitochondria. Right? What's going in is pyruvate. And I'm sorry, I'm trying to use your chart, but my handwriting is awful, so I'm trying to write big as well. So pyruvate comes out as acetyl CAA. Right? We also have NAD plus coming in, and it's going to go out as NADH. Okay. So that's what's happening here. We don't get any energy, but we're now ready for the Krebs cycle. All right, so this is a good stopping point if you need to go back or fill in your chart, but I'm going to move us now to the Krebs cycle. All right, so again, we have this complicated set of reactions, and notice it's a cycle. Okay, so at the end of this process in here, we have to regenerate our starting molecule, right? Anytime you have a cycle, that's the whole point. You want to regenerate the starting molecule so that the next thing that comes in can bind to it and the cycle can keep going, okay? But again, don't worry about the intermediates, okay? Just overall look at what's happening here. Now, at this point, remember that at the end of glycolysis, we ended up with 2 pyruvates, right? And that's important to note. The 2 pyruvates is important because what this is showing you is only 1 pyruvate going in as acetyl CoA and going into our reaction here. But actually, the Krebs cycle is going to happen 2 times because you have 2 pyruvates, right? So for every sugar, you're going to do this process 2 times. So when we're totaling, and I should have on the last slide for that little half step, we really ended up with 2 NADHs because that half step took place for both of the pyruvates as well. So you, from glycolysis onward, you have to double everything because for each sugar molecule that enters cellular respiration, you're going to end up with double the amount. All right, so this is also called the citric acid cycle because one of the first things that you create is citrate or citric acid. Some people call it the tricarboxylic acid cycle or something like that, or the TCA. TCA, citric acid cycle, Krebs cycle, it's all the same thing. So what happens now is our little acetyl CoA goes in and attaches to a molecule, and it's going to go through a series of transformations. Notice carbon dioxide is going to be coming off, so we're losing that waste product of cellular respiration, right? But also notice 1, 2, 3 NAD pluses are going in and coming off as NADH. So we've got 3 NADH that are produced. Here comes a new friend, FAD, that goes in, and you get 1 FADH2 that comes off. Just another hydrogen carrier, or electron carrier. It carries electrons and hydrogens, right? But also, in the process, right down here, right here, notice we're going to generate an ATP. Remember, this is substrate-level phosphorylation, so it's really not that exciting. You're only going to get 1 per cycle, so you are going to make an ATP in this process. And then again, at the last step, you're going to take this oxaloacetate and we're going to convert it, you're going to take this malate and convert it to oxaloacetate to regenerate our starting molecule. Okay, those are the important features here. The one thing to understand about this process is this last bullet point here. By the end of the Krebs cycle, glucose is gone. Okay, we're done with glucose. But there's still one more step, and that's really interesting. How is it possible that we can still generate energy from glucose even though we're actually done with glucose? The answer is here, right? In the previous steps, we generated like 4 NADHs, right? In this step, remember, this is times 2, this is times 2, and this is times 2, right? So here we've generated 6 NADHs, we've generated 2 FADH2s. So we've got a whole bunch of these electron carriers now. Those electrons are carrying the energy from the original glucose molecule, because once we broke down the glucose and moved the electrons from glucose over to these molecules, the energy came with it. Okay, so now what we're going to do is we're going to end up going into the electron transport chain, and here is where the energy is in this molecule and this molecule and the ones that we generated before. Those are still carrying glucose's energy, and that energy is going to be used to generate mass quantities of ATP, because once we're done with this, we're going to do a new kind of phosphorylation. All right, so let's fill in our chart. All right, so for the Krebs cycle, we're in the matrix, right? What's going in are, and I'm going to put the doubled amounts here. We're going to do 2 ADP going in, and those are coming out as 2 ATP. Right? Oh, the first thing that goes in actually is the Acetyl CAA, right? And that's actually coming off as CO2, right? And I'm not going to, because CO2 is a waste product, I'm not going to worry about the amount. You do have to remember, though, that there are 2 acetyl CoAs, which means you're going around the Krebs cycle 2 times, right? Which is why I say that we actually end up with 2 ATP. Each turn around the Krebs cycle, you get 1 ATP, right? All right, we end up with 6 NAD pluses going in and 6 NADHs coming out, right? You end up with 2 FADs coming in, and they come out as 2 FADH2. Okay, now remember, we had, I think, 2 NADs here, NADHs here, and I can't remember glycolysis if that was 1 or 2. Let's just put, oh, it was 2, because I think it was 1 per, I can't remember. Anyway, we got some NADH here. Whoops. And then we've got this NADH and this FADH2. So while the energy has been all impressive, or actually unimpressive, we haven't generated all that much energy from this process, we have generated a bunch of these energy carriers. All right, last thing, the electron transport chain. This is by far the most complicated part of the process, but it is the most important, because this is where the money is, okay? This is where we're going to get the ATP, right? So this is really important. What happens is our molecules of NADH and FADH2 are going to be dropping off their electrons to various molecules that are embedded within the inner mitochondrial membrane. You don't have to remember what each of these molecules are, but you do need to know that these are molecules that are waiting embedded in that membrane of the inner mitochondria, of the inner mitochondrial membrane, and they are accepting electrons from our electron carriers, okay? And it's a transport chain. See the word transport here? So what they're going to do is they're going to pass the electrons from here to here to here to here to here. And as the electrons pass, notice this graph is showing you the energy, the free energy available in those electrons as they're passed, okay? I like to describe this process as like a magic trick, because when you're doing a magic trick, there's the trick that you see the magician do, but then behind the scenes, there's some sort of sleight of hand. And it's like that in this process, because most of us have our attention focused right here, right? Electrons, what's going on with those electrons, right? But the actual important thing that's going on is what's happening to the hydrogens that were forced to give up their electrons, right? The way that the electrons were transferred is in this little hydrogen that's attached to these molecules. And we're going to need to pay attention to those too, because it's the movement of those hydrogens that's going to generate ATP for us, okay? But overall, that's what's happening in this process. And again, remember, we didn't use oxygen in the pyruvate processing half step. We didn't use oxygen in the Krebs cycle, but we will use it here. Okay. So overall, that's what's about to happen. And so far, all that I've showed you is what the magician wants you to see. The electrons being passed and the oxygen being the final electron acceptor at the end of this process. All right, so far in the electron transport chain, remember we're on the inner mitochondrial membrane, right? And we're going to drop off all of the NADH, and we're going to drop off all of the FADH2, and those are going to come off as NAD plus and FAD. FAD. All right. The other thing, though, that we're going to bring in is oxygen, and it's going to come off as water, and I'll show you how in just a minute. Okay. And then finally, we're going to bring in 36 to 38 ADP, and that's going to come in, or sorry, the total ATP is going to be 36 to 38 for the whole process. Here, actually, we are bringing in 32 to 34 ATP, or ADP, and we're going to generate ATP from each molecule there. Okay, so we're going to generate 32 to 34 ATP. I'm telling you this ahead of time so that you can look for it, because what I want you to remember is the point here is to generate a bunch of ATP. Okay, and what you can see is we generate a lot of ATP here. All right, so here's the electron transport chain. And again, here what you're seeing is the electrons coming in and being dropped off and bounced around, right? And as they're dropped off, they get bounced along down this chain. That's all well and good. That's the front of the magic trick. That's what you're supposed to see. But notice, NADH becomes NAD+. It's not just the electron that's removed. You're also removing this little hydrogen ion, and the hydrogen ion gets pushed to the inner mitochondrial membrane space, okay? It just gets pushed out. Notice it's getting pushed out here and pushed out here, okay? That's the sleight of hand behind the scenes, okay? And that's important. What we're doing is building up a concentration gradient of hydrogen ions right out here. So while these electrons are being passed along, losing their energy, blah, 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 everybody's focused on that. Ultimately, what's going to happen is you build up this concentration gradient, and we need a concentration gradient to push this guy into action. ATP synthase. This is the enzyme on steroids. This is the best, strongest enzyme that has ever existed. It's like a little motor, okay? What happens is we're building up this concentration gradient because we want the hydrogen ions to be forced to come through this ATP synthase. Because as they do, they crank this little motor. And as this motor cranks, the energy is used to take ADP and phosphate and phosphorylate them together into ATP. Okay, and that's the key. So while, yeah, we're paying attention to the electrons, woohoo, the electrons, what's really important here is the concentration gradient, because that's going to fuel, that's going to give us the energy to crank our ATP synthase enzyme, and the energy from that is going to be able to generate the synthesis of ATP. Okay, that's what's important here. Ultimately, in the end, some of you are saying, well, wait a minute, that's going to put a bunch of hydrogen ions back into the membrane. Yes, it does, but look who's willing to accept those hydrogen ions, right? Remember I said the electrons are accepted by oxygen, right? But they're also, the oxygen will accept the electrons, but it's also got to accept these little hydrogen ions, and that's why the oxygen plus the 2 electrons plus the 2 hydrogen ions generates water. And we're not worried about how many water on your sheet, because it's a waste product from this process. Okay, so we've generated a whole bunch of ATP now in the process of doing the electron transport chain. All right, so let's get to that. That whole thing that you just saw was aerobic cellular respiration, right? So that's aerobic cellular respiration. What happens if there is no oxygen present, right? We do glycolysis, which is what I told you before. Remember on our chart, we put a red line after glycolysis and said if no oxygen is present, we can't go through pyruvate processing, we can't go through the Krebs cycle. Instead, what we do is go through fermentation. The whole point of fermentation is to regenerate the NAD+, right? In glycolysis, we created NADH, sorry. But now we don't need the NADH anymore, right? There's no oxygen. We don't need electron carriers because we're never going to proceed to an electron transport chain. And so the whole point of fermentation is not to so much to make the alcohol or the lactic acid that you guys learned about in baby bio. It's about taking that NADH and stripping it of the electrons and hydrogens so that when oxygen is available, then we can go ahead and use the energy from glucose to reform NADH later. There's no need to build up NADH in cells that don't have oxygen in them. Okay, so there's the point of fermentation. All right, so what we do here, this is glycolysis at the top right here, and we've generated NADH. But now what we have to do is we have to ferment this pyruvate, and that allows us to strip it of the hydrogens, put them back on the pyruvate. Now in plants and in yeast, you're going to get alcoholic fermentation. In other words, the end product here that you get is ethyl alcohol, right? So when we put the hydrogens from NADH back onto the pyruvate, we create ethyl alcohol. In bacteria and animals, what we get is lactic acid. Okay, so lactic acid fermentation is called that because its end product is lactic acid. Alcoholic fermentation is called that because its end product is alcohol. All right, couple more things before we leave this topic. I know this is long, just bear with me. This is the baseline reaction for pretty much every other reaction inside of your body. So while we think of this as a process that we use to get the energy from glucose, you can actually use this process to build other molecules, but you can also use this process to metabolize things besides sugar. So the first thing I want to look at is what can we put into this reaction to metabolize? Notice, here's our steps, right? Here's glycolysis here, right? Oh, whoops, here, glycolysis, right? Here's the Krebs cycle here. Here's the pyruvate processing, right? Notice we can put fats, we can break them into glycerol and fatty acids, and the glycerol can be broken down in glycolysis and then go through the steps. Fatty acids can be converted directly into acetyl CAA and then go into the Krebs cycle. Proteins can be broken into amino acids and can go in as pyruvate. They can go in as acetyl CoA, or they can go in at the Krebs cycle. So when we talk about metabolizing other things besides carbs, and even other carbs like starches and things like that, they're going to get broken down into their little carbohydrate subunits and enter at the beginning of this process, right? But proteins, fats, they come in and they can actually enter the same process, cellular respiration, but at different points and still be metabolized. So that's how we get energy from other biomolecules. Finally, I mentioned also that this is the baseline process that we use to produce other biochemicals. So you can actually use this for anabolism, not just catabolism. And what this is showing you is you can actually siphon off some intermediate molecules at various points to produce other things. So glucose can be siphoned off from one of the steps of glycolysis to create RNA and DNA, right? The acetyl CoA can be siphoned off to create fatty acids that can make fats for us. They can also make phospholipids for our cell membranes. A lot of steps of the Krebs cycle, you can siphon things off to make amino acids that you can use for proteins later on. So this process of cellular respiration, remember, it's versatile. You can use it to break down lots of things. You can also siphon off or pull away some of the intermediate molecules that are in the process to create other things. That was a mouthful. That was, I think, a 30-minute video. Thank you for your patience. Make sure to text me with questions.